So let me start by introducing myself a little bit, give you some background. I am a professor in biology department at Temple University. I also have an appointment in computer science, and I've been here for 7 years. And I also, at Temple University, direct the Institute for Genomics and Evolutionary Medicine. Some of the topics that I will be discussing today relate to that directly. And just by my background, to give you an idea, I have degrees in engineering and biology from India, where I did a double degree early on. It was somewhat unusual choices available at the university I went to, BITS Pilani, BITS Pilani. And I was always fascinated by biology and, of course, very interested in engineering. During my PhD, which was at Penn State, I combined the two areas and studied how genes and genomes change over time. And it was a lot of engineering knowledge that I had of problem solving, coupled with the need to make observations in biology, formulate hypotheses, and test hypotheses. There's a combination of two that I do in my research. And if you want to know a lot more about my research, kind of articles I write, kind of things my group does at Temple University, you can look at the first URL there, this tinyurl.com at Kumar 2021. It gives you an idea of a brochure from my group, which is a abbreviated summary of what we do and some cool things we do, and also more extensive information about our group in kumarlab.net. And the pictures here you see are pictures of some students and postdocs in my group that are currently working with me. So I have 10 of them. So it's an excellent group to work with. And I have, of course, some other technical staff members too. So the area that I am very interested in is genetics and genomics, which has to do with our DNA, for example, as well as the genetic material of pathogens. And genomics is a study of really genomes as a whole, understanding genome function, and also use genomes to understand biology and diversity of life on Earth. And my focus during my PhD work was evolution and genomics. So in evolution, you try and understand what happened in the past. And the past could be quite recent, like you and your siblings and your cousins and your family, extended family, followed by your geographical region, your religious, ethnic, personal group, all the way to populations, up to species. So I'm interested in that kind of history, tracking it, using genomes, and studying the genomes of these individuals and species. So this is the area of evolutionary genomics, or evolution and genomics. In the last couple of decades, I have become very interested in combining this with medicine, because in medicine, we are interested in well-being, treatment, health of individuals. And really, every one of us is a byproduct or a product of evolution. We have come from our parents, but with modification, where half our genome is from mother, the other half from father, and both mother and father DNA underwent change because they need to copy the DNA to be inherited to us. So essentially, this is our mini or personal evolution. But if you go back in time like this, you can build pedigrees of individuals, populations, species. And because we are all connected in that way with our ancestry, in medicine, many times people from the same family respond similarly to different drugs, different diseases. And so that's why evolution and medicine are highly intertwined now. And genomics is the reason for that. And also we know that many diseases happen because of genomic changes or mutations. And we also know that many protections sometimes we have from diseases due to genomics and medicine. So this is the area that I work in, evolution, genomics, and medicine interaction, which I will talk about with you today. And so just to give you very quick definitions, genomics is a study of genomes, whether you want to use genomes and their function study, or whether you want to use genomes for study of other aspects. Medicine is, of course, the practice and science of diagnosis, treatment, and prevention of disease. And prevention part is becoming more and more important. And evolution has to do with change over time through natural selection and through random chance event, like mutation and its fate over time. And you've heard a lot about Darwin's natural selection. We also have similarly neutral theory of molecular evolution in the analysis of genetic material. So these are things, some of them are advanced topics, and you will learn more about them in college. But this is the areas that I work in: genomics, medicine, and evolution. And my group is very interested in the study of mutations. And mutations are basically de novo changes that happen in a DNA or other genetic material of a cell. And usually they are inherited. Then they are called germline. That means these are the mutations that occurred in a sperm or an egg cell, which then can be inherited or passed on to the offspring. Not every mutation is passed on to the offspring, because generally a sperm and an egg, only for an individual to become an embryo, to form an embryo, we just need one sperm and one egg in the case of humans. So germline mutations are the ones found in those sperm and egg cells of mother and father in our case. Then there are other mutations that happen in our body outside of the sperm and egg cells, which are called somatic variants, somatic mutations. They happen in tissues and organs. And the greatest example of somatic variation is, as you know, various cancers, which are a product of a lot of somatic evolution or somatic change over time in a person. And so we also study that because it's about mutations and variation. Basically, mutations and in germline create population level variation, and mutation in somatic cells, non-germline cells, create within body variation. And in our field, in evolution, mutations are considered the raw material for change, and they occur in every organism, and I think in every cell now. We know even in a body, no two cells are identical, because there's always some difference between them, because they're coming from a copying of DNA, in our case, for example, from other cells. So mutations are really everywhere. The mutations happened in the past, billions of years ago, when the simplest cells were formed, and they've been happening since then to now, recently, hundreds of thousands of years ago. And mutations are happening at present in families, like you and your sibling will share much greater similarity in your DNA than you will with your cousin. And this is one of the ways in which, in paternity analysis, they can tell who may likely be the father, or in this case, mother also you can find. And mutations also happen during the lifetime of an individual, as I mentioned, which are, for example, many times leading to tumors. Not always, very rarely, but it does happen. And so these are the mutations we're interested in studying. So mutations are a way for us to reconstruct our history and history of ourselves, both also, because it is almost as if the record of change is written in genomes, which can, when we compare them across cells in a body, across population across individuals in a population, across species in a biodiversity zone, and across all species on Earth, to understand evolution on Earth. So basically, mutations are very, very useful. And future, they will always occur. And coronavirus, for example, you hear about a variant, new variant came. Coronavirus also mutates, like humans mutate. It mutates faster than humans, but it will constantly change. Similarly, when you have offspring, they will also have mutations, and we have mutations currently. So it's just constantly happening. So what do we do in my group? In my group, what we do is we take these big data sets of species information, and many species genomes have been sequenced, like hundreds and hundreds of mammals and lots of birds, all kinds of flies. And we build evolutionary trees from that, because there isn't enough fossil record to tell us relationships or when they emerged. So this on the right side you see is a time tree or phylogeny of species A through G, with A and B showing a very short time of divergence, and they are much closer to each other than either one is to C or D. So this is the kind of time tree we build. Also, you can build phylogeny of pathogens, and I'll show you an example today on coronavirus. We also develop methods for fast analysis. So we build evolutionary histories. For that, we have large amount of data, and you need computational methods. It is not trivial just to do that by hand. And so you write software, you have to develop new algorithms, new statistical methods for diagnosing mutations, also tracking metastasis in a patient. If a patient has multiple metastatic tumors in, let's say, lungs and kidney and brain, what was the movement? Where did it start? We can discover that through phylogenetics. And then we develop resources to enable discovery. So we also write software and databases that are useful for you. So I would simply say that building the time tree of life is a major project in the lab, and we have a resource we have built. And in this building the time tree of life project, we are trying to really tell when different species have a common ancestor, at what time, how many millions of years ago, for example, did humans and chimp have a common ancestor? How far ago do we have an ancestry, or primates have an ancestry with birds? So this is the database that we are building. And if you by chance watched Rise of Animals series by David Attenborough, they actually used our time tree to illustrate evolution of mammals and other species in their TV program. So these are generally useful for students, and it's a fundamental knowledge in biology. If you want to experience time tree, you can go online to this website, our website, and you can press in dog and cat. And if you do that, it'll give you a time information and say, well, dog and cat likely had a common ancestor around 50 million years ago. So this, similarly, you can ask humans and chimpanzees, gorillas and orangutans, and it will give you knowledge that is otherwise stuck in literature, which are hard for anyone to read beyond the experts. So this brings all the expert knowledge to you at your fingertips. So give that a try if you're interested in this sort of a topic. And also, you can build a time tree of, for example, primates by asking, hey, give me a tree of primates. It will show you a time tree of primates. So this is what you can do on the timetree.org website, which is useful for you to learn about biology and also experience real biological literature. For the mega software, your teacher and Joe told me that you guys will be doing a exercise later in the week. So mega software I developed as a graduate student in 1993 first. Now we are in the 11th release that has just been put online as a beta version, but I think you should use that one potentially for your class. And essentially here you can actually give it sequences and build phylogenies. Here I'm showing you a phylogeny of influenza in different animals. Here swine, which is from the pig group. And essentially they were sampled on different times over the years. So using phylogenetics, you can build phylogeny of these viruses, for example. So what I will do now is going to discuss with you the pathogen phylogeny, just to give you one snippet of my research, very recent one. And I know you already learned a lot about coronaviruses in a previous class from a medical perspective. And what I will do today is to give you an idea from the evolutionary perspective. What do we know? And maybe some of these things will resonate with you regarding the recent knowledge that you have acquired. So this is COVID-19. And you know, COVID-19 was a new disease that was first discovered in December in Wuhan province, Wuhan in Hubei province of China. And it's called COVID, as you might know already. CO stands for corona, VI for virus, and D for disease. So it's COVID. And it was first found in 2019, so it's called COVID-19. It is kind of a nomenclature often used by virologists just to give names. So this is the virus that began in 2019, or was first found in 2019, this disease itself. And why is it called novel coronavirus? Because it was new at that time. And now, of course, it has become relatively old news, ongoing. And this COVID-19 is a new disease because it is very different from other coronavirus diseases, that cause common colds in humans. So COVID-19 is a disease that goes beyond that and actually is much more detrimental to our health and our population. So, this is a new disease with a very different set of symptoms and extremely deadly outcome for many people. But important thing to remember is there are many pathogenic coronaviruses for humans, and a huge diversity of coronaviruses exist in the population in various species, including bats, including camels, including other mammals, and humans for that matter. Most of these have very mild symptoms in humans. But there are a couple of major exceptions. One is SARS, that was almost 20 years ago, which was found to, after many years of research, to go from civets into humans in the Chinese Guangdong province, and ultimately more than 8,000 infections were recorded, which is from today's standard almost nothing, and less than 1,000 deaths, and now we have lost 1 million people to the newest disease. The other one that happened more recently, 10 years ago approximately, is the Middle East respiratory syndrome, which actually began, it's called Middle East because it was found there first, and it began in Saudi Arabia from camels. So this is a zoonotic origin. From an animal, we get this disease. And so camels transfer this disease, these viruses, to humans who are living in close proximity with camels, which is very common in Saudi Arabia and in countries of that region. And it also has less than 1,000 fatalities. So again, this particular group of viruses are known to create problematic lineages, evolutionary offshoots, but they never had as bad an effect as did coronavirus. And then how do we tell a relationship of these viruses with coronavirus that we are dealing with now? We build a phylogeny. And the phylogeny we learned early on that SARS-CoV-2 in this red color, in this blue box, that genome was sequenced from the patients, because when they took the patient sample and sequenced all the genomes they had, they found that they had the coronavirus. And this coronavirus, when they compared to other coronaviruses they knew that existed before, they found that it was closest to the bad coronavirus that was sampled in the same region in China, similar region, not Wuhan, but far away from it, but in a similar region. And they were very, very similar, really 99.6%, 96% similar actually. So they are very, very similar. On the bottom here is 0.1, which you can sort of translate to 10% or 8% dissimilarity. But then this told us about the potential origin of this coronavirus in causing COVID, that it may have come from a relative of or from this bat that contained this virus, its closest relative. It's not known yet where it came from, which animal, or directly from bat to humans. But then they realized that this coronavirus, SARS-CoV, is in fact very similar to original SARS 20 years ago. You can see SARS-CoV, and that's why they named it SARS-CoV-2, because it was so similar to the previous SARS. So 20 years ago. So the 20 years ago, one lineage now had a common ancestor shown by this open circle here, which gave rise to COVID-2. And so this virus evolved from the same parent from which the previous SARS virus evolved. But if you look at MERS, go down and look at another red colored name here, MERS-CoV, 10 years ago, this was the epidemic. And you can see that this particular COVID is very different because it is far away from COVID-2. So a phylogeny gives you this information. And this is the area that we work in, developing new methods and developing new software. But in addition to all three of these, you can see some orange color coronaviruses. And all of these coronaviruses are actually well known to cause common colds in humans. And so you can see the cold coronavirus is found in the same group as a whole as is the deadly coronavirus we have. So different coronaviruses have different attributes. Like in this one, spike protein in COVID-2 is very complicated and difficult for us to deal with. But coronaviruses will constantly be coming, unfortunately, and causing problems. Sometimes there will be serious problems, and other times there will be not so serious problems. And so this gives you an idea. And then these solid circles here indicate that we have a lot of confidence, more than 90% in these relationships, as compared to the gray circle, which means some rearrangements are possible. And then what we recently did is we said, well, what is the history of COVID-2 in humans in the last 15 months since we have known coronavirus? And so we built an evolutionary tree of our coronavirus disease, COVID-19. And so in this one, what you are seeing is all these little offshoots starting from what we call the progenitor or the parent coronavirus. And this parent is probably originated around 2019, October to August. And it gave rise to variant V1 and V2, and alpha 1, alpha 1A, alpha 2, alpha 3. So you can see these are all variants. So you hear about variants all the time today. These variants have been occurring all the time in the past since the parent coronavirus existed. And this could be the first patient who had a coronavirus that could spread it around. And as you can see, if you go down here, we find that the Wuhan 1 coronavirus is actually this coronavirus on this lineage. So Wuhan, where we had the outbreak in the market, actually had the coronavirus that came from a parent coronavirus that existed for some time in the population. And that coronavirus gave rise to many strains which traveled the world, because new 1, new 2 are found in the USA in January 2020. And then many of these were found right away around that time in Europe and other countries of Asia. So already this coronavirus, much before December 24th, was worldwide spreading. We only saw these variants when we sampled them in January last year or February last year, but they actually likely came from the parents' early offshoots. And in December
Dr.
last
Kumar.
year, yes. Go ahead.
Sorry, I have a question. So there's two terms on here that that doesn't mean period's over. Sorry. So there are two terms on here that I see, and actually I will eventually teach them, but they might be helpful, is synonymous mutation and non-synonymous mutation. Can you explain that?
Yeah, so basically, in the genetic code, we have DNA, and DNA is then used to make protein. So essentially, this is called the process of translation. You translate from a triplet codon-based sequence of RNA into an amino acid sequence, which is ultimately the protein. And whenever there is a mutation that does not change the amino acid encoded, then it is called a synonymous change. And when a mutation changes the amino acid encoded, it is called a non-synonymous change. And you can see in the genetic code table, there are 64 cells, and there are only 20 amino acids. So multiple cells correspond to the same amino acid, and therefore same residue, and therefore that is the distinction between synonymous and non-synonymous. If a mutation does not change the meaning of the codon in terms of amino acid residue, then we call it synonymous. And as you can see, there are green branches, and then there are red branches. So red branches refer to non-synonymous changes, and green branches refer to synonymous changes. So during the history of coronavirus, many non-synonymous and synonymous changes occurred throughout. It's not only non-synonymous. And sometimes synonymous changes have no effect on function, and often non-synonymous changes have an effect on function, but it's very hard to tell without experimental laboratory work generally. So essentially what this picture tells us is that the Wuhan outbreak was not the start of the COVID-19 virus. The start was much earlier. And lots of variants existed before December 24, 2019, which was the first time we had the sample of coronavirus in China, because their children, their offshoots are detected later on in January 2020. And that since the Wuhan virus was discovered, there have been a lot of changes, as you can see. A lot of variants have come out. And even now today, variants are coming out the whole time. If I build this tree today, expand it, it will show you even more variants. And I'll talk about some. So when we start to call some variant as coming from Wuhan at the start of the pandemic in Wuhan, the virus itself has a longer history when it comes to even the virus that causes SARS-CoV, COVID-19. It's not simply that it all began in a laboratory in Wuhan and spread. Now, entirely possible that all of these variants, could have been found in the Wuhan area at the time, but we never sampled enough genomes to find them. But the point is that their offspring are already present in January, so we know that they existed. This is the Wuhan one. And then just to give you an idea of a simplified view, you have the progenitor or the parent COVID. It underwent multiple mutations, which we call alpha ones and new ones. And then earlier samples from 2019 mapped to this particular branch. And then there have been a lot of variants produced, as you can see. And as you can see, that most recently in the news, you hear about London variant and South African variant. They actually happen to be the same change, N to Y in amino acid residues. And these changes are actually, they have occurred independently in London and in South Africa. And these ones make the vaccine less effective, South African strain. But London one is still responds to the vaccine well. So coronavirus is constantly undergoing variation. And remember, South African strain did not arise in response to vaccine. Vaccine is just arrived in South Africa. So this variant was just naturally occurred or happened because of random process of mutation. So this gives you an idea that using evolutionary biology and genomics, you can learn a lot about the disease cause, which is COVID-19. And I'm going to just show you quickly that if you look at Asia and 2020 up to 2020, end of 2020, this right here, that the green zone, these green variants have spread a lot, and the orange became less and less common, which is alpha type and epsilon type. Then if you go to the next one, Europe, green were very common, but suddenly they were overtaken by eta ones, you can see. And in Europe, we started to sample in February, so we don't really have data from before. But as you can see, the dynamics is different than Asia. And in North America, in our country and Canada and others around here, we didn't start to sample until February either. But you can see we have the blue one, which is Delta, and epsilon is less. So different variants by chance spread around more in some places. It doesn't really mean that one variant is more adapted or has a greater or less effect. It by chance certain variant types will spread more.
So we have
Go
a
ahead.
question here. In describing the graphs that you're seeing here, is the black line representing the number of cases?
This is the number of genomes, yeah.
Okay, so the lines that doesn't have a color related to it, just the actual line, that's the number of genomes.
Yes, basically whenever in an area people sample genomes, then they put it in a database online, and then we retrieve the data. So the number of genomes will vary a lot in different parts of the world, because, for example, in China, they stopped the pandemic completely, right, in February or March. So we have no data from China after February, basically. In the US, we had a great decrease in the pandemic, so we have no data. Then when the pandemic increases, there are more data. So this is why you will see a big variation in this count. Because this is actual number of genomes of the coronavirus, not of the patients themselves. Because as the number of patients are fewer, when we take their sample and generate the genome sequence, we rarely get them. And so that's why you will see a big change. In Europe, you remember they had curtailed the problems for a long time, and suddenly new countries in Europe got infected. So whenever a new country gets infected and it spreads in that country, this happens. So if you look at the US and different states, you will see very different patterns, but New York and LA versus West Virginia or Colorado. So it's very interesting. These patterns of number of genomes is very variable. Okay, so does that help?
Yeah, I think so. Does that?
Okay. Yeah.
Yes.
Remember, there is no. These are just independent scientific laboratories that are sampling individuals and sampling their coronaviruses. So there's a tremendous heterogeneity over time because of the number of genomes that are there and the number of countries that are participating. And some countries don't spend any money on acquiring data. So it is highly heterogeneous. But you can see the relative proportions are given here, which I think you can see consistency in. Because even if there's a sample of 100 or 200 genomes from September or October, you can actually make a pretty good estimate. So you don't need thousands of samples in general. Okay, so I wanted to just end by talking about a new degree at Temple, which might be of interest to some of you. The Temple has begun a new degree in genomic medicine. So like you do a degree in biology, where your primary research area, your primary interest area is in biology, we actually have a degree now, if you like to do, in genomic medicine. And this is really a superset of biology. So what is genomic medicine is, that genomic medicine will be the next generation of medicine. Whether you actually go be a doctor or be a researcher or somebody interested in cancer, disease like diabetes or Alzheimer's, they have a genomic basis. And genomes tell you the ultimate cause of disease in many, many cases, like the genomes of the pathogens or genomes of humans or hosts. And personalized medicine, as you know, is a big deal these days. So what we did at Temple is to create a curriculum that trains you in biology plus genomic medicine. So you get to do a few extra courses as a part of your electives, which are required courses for genomic medicine major. So when you come out, you are actually equipped with not just biology, which is great and important, but also have a greater appreciation of genomes and medicine. So the interest is in developing a thorough understanding of genome and its role in disease, prepare for research and or medical careers focused on genomic medicine, which could be genetic counselors, you could work in research institutes, or you can be a doctor doing an MD or a PhD MD in this area. So that's what you can do. You will be at the vanguard of medicine or medical revolution. And interesting about genomic medicine is we give you enough information that when you go and go to a master's degree, go to a job elsewhere, go to MD, you would know about biology like you need. Medicine is add-on. Things about data, because the future doctor is going to be a data-driven person rather than simply remembering what they knew and listening to other individuals that are, in the drug companies, pharmaceuticals. In fact, many pharmaceutics would be genetically marked, so individuals with certain mutations can take them or not take them. You will learn a little bit about computer programming, not a huge amount, but enough so that you will be able to wrangle data, and you will study evolution because your ancestry of the patient is extremely important. Some patients can drink milk because of their ancestry. Some cannot, for example, drink beer because they come from parts of the world in which there is no tolerance for beer in the genetic makeup of people. So you have to know the genetics for this person. So what kind of courses do we have extra in addition to biology courses you do? Genomic Foundations of Medicine. You will actually learn about disease, genes, and mutations, which is very important if you want to have a career in which you're interested in helping people through research or clinics. Pathophysiology of genomic medicine. How do genes and mutations affect your cell and physiology. So we want you to be aware of this before you go to the next career or MD career in life, and also how to understand evolution, how to have a better appreciation of evolutionary biology in the context of medicine and medical phenotypes or traits. So essentially, you become a very well-rounded person entering an MD, PhD, master's, or regular workforce. And so that is our interest in making you well aware of these things. And there is a pre-med track and regular track. And remember, genomic medicine is going to really strongly inform you about everything you could do. And then you can make a better choice. This should also be more competitive in medical schools to get into medical schools, because hundreds of thousands of students are biology majors. What makes you special? Part of it is the degrees you do, and it should be much more interesting for a medical school to have somebody with genomic medicine degree than a degree in biology without real emphasis on something. So it's not for everybody, but it is, I think, something that can help a lot of people. If you have interest in genomes of humans and pathogens, then this is a degree you should think about and explore further. So I hope that helps. Thank you for your attention. I'll be happy to answer any questions.
Again, anyone online who has a question, you can use the hand raise function. All right, we have one. You can just ask Krish.
Oh, okay. So first of all, hi, Dr. Kumar. And my question is that I have a bunch of family friends and cousins majoring in computational biology. So is genomic medicine similar to that?
So computational biology major probably is a master's major, because, but generally computational biology can be about many things. You can do it in ecology, environment, medicine, genomes. And so this major, if you plan to go into the field of computational biology after your undergraduate degree, would be perfectly positioned, because you would have learned about some programming, some data, and a lot about genomes and medicine. So you will be really ready to go into a master's or a PhD in computational biology, and even in a company, if you already program and stuff, to go into that company and work there after your degree.
Okay, thank you.
All right.
I think we had one more person. Maybe Krish had a question, but maybe the same one.
That was Krish. Yeah.
Okay.
All right. Well, I don't see any other people here that have any questions, so I'll let you get off to your vaccine appointment.
Yes, right.
And
I have to go to a vaccine appointment and get the second shot.
All right. Well, thank you very much. I'm sure all the rest of the students are very appreciative of your time, as I am, and thank you very much.
Thank you, everybody. Enjoy and good luck with your future